I cloned a $20,000 exotic cat in my DNA laboratory. Here's how it happened. I was in my lab cloning my brother's wife when I suddenly thought I needed to clone something cooler. So I left the lab and headed straight to Rodeo Drive in Beverly Hills, California. When I arrived, I searched everywhere, right, left, every designer store, every single street. Then, out of the corner of my eye across the street, I saw it. A miniature cheetah-looking cat with a flawless golden coat. I realized it was a Bengal cat. How was I going to get the cat's DNA without getting caught? That's when I called up my friend Ashley. She said she only lives 5 minutes away. I waited. She finally showed up and I told her the plan. She walked up and started talking to him. I snuck up right behind them, leaned down, and swabbed the DNA directly from the cat's mouth. Sealed the DNA swab in a chemical grade tube and vanished. That's when the cloning process began. First, I treated the swab with red lysis solution to break down the saliva protein. Then, I spun it in a centrifuge to isolate the DNA. Under the microscope, I examined the strands, pure, unbroken, and alive. Hours later, cells started forming. Now, this is him at 9 weeks old, and soon I will have a $20,000 exotic cat for myself.